on to Shan Luo from Hong Kong, who's presenting with her.
Good afternoon, everyone. I'm Shan from School of Public Health, the University of Hong Kong. And thank you for the organizer for providing me the great opportunity to present our recent findings that metformin could promote healthy longevity based on genetics, which is recently get published. So the global demographic have shifted from rapid population growth to the population aging. Hong Kong has the longest life expectancy in the world. We are living much, much longer, but are we living healthier? As a public health practitioner, we are looking for sustainable strategy to promote healthy longevity. Metformin is the first-line medication for type 2 diabetes. It is highly affordable and safe. It has been listed on the WHO model list of essential medicine, and it is increasingly recognized it has the potential protective effects against age-related disease, and has been proposed for healthy longevity. And I think like Dr. Neer have convinced everyone that metformin is the most suitable drug for testing healthy longevity, and the groundbreaking TAM study is in its preparatory stage. But given the limitation in the conventional observational studies, such as the confounding and immortal time bias, and in the absence of convincing evidence from large-scale RCT, and we aim to use the population genetics to provide kind of evidence that metformin's potential in healthy longevity. And the genetics have already contributed to the proof of concept trial. For example, the PCSK9 for coronary artery disease and IL-6 for rheumatoid arthritis. And our previous study using Mendelian randomization designed have provided the evidence that metformin have its potential protective effects against the aging-related disease, including cardiovascular disease, cancers, and Alzheimer's disease. So the fundamental question, whether it can be repurposed for healthy longevity, and we have used the multi-omics data, including the tissue-specific gene expression from GTEx portal, and also the genetic and the phenotypic data from the UK Biobank. Metformin is very complex drugs with multiple mechanisms of action, and although we are not fully understood, but these are the main pathways have been identified. The AMPK-dependent mechanism by reducing hepatic glucose production and the lysosomal AMPK pathway, PN2, induced by low-dose metformin. And in addition, the AMPK-independent mechanisms such as inhibition of myocardium GPDH and resulting improved energy balance and reduced myocardial stress. And we systematically searched the literature and the drug bank. We identified 4 putative protein targets of metformin and in varying 10 genes. So in a Mendelian randomization study, we used the genetic variants randomly allocated during the conception, which is analogous to the randomized procedures in an RCT, which makes Mendelian randomization study is less susceptible to confounding and reverse causality. And it also relies on instrumental variable assumption. And first of all, the genetic variant should be within or near the protein encoding gene, and it should be associated with the non-downstream biomarkers. And it can be selected on the basis of their association with gene expression, protein expression, or the non-downstream function, such as metabolic levels of biomarkers. And secondly, the genetic variant should not share unmeasured cause with the outcome. And lastly comes to the concept also known as exclusion restriction. The genetic variants do not affect the outcome except through its effect on a protein target, which exerts its action through the downstream biomarkers. So we assess these assumptions by selecting the genetic variants with evidence of gene expression on the HbA1c using Mendelian randomization study. And we also conduct genetic colocalization to make sure the gene expression and the HbA1c are sharing the causal variants. We restrict our analysis to white British participants in the UK Biobank and adjust for the top 20 principal components of ancestry to control the population stratification in the UK Biobank. Although the exclusion restriction assumption could not be fully verified using data, and we also conduct sensitivity analysis using methods that relies on different assumptions. So a variety of aging matrix have been introduced in the germ science framework, and there are lots of aging craft developing. So here comes to another essential questions, like what kind of endpoint should be used. And consensus may be difficult to achieve, and one of these is the blood phenotypic age. In addition to the chronological age, the algorithm also incorporates the 9 multi-system clinical biomarkers to predict their phenotypic age, and it can tell a person if physiologically younger or older than its own chronological age. And also, the telomere is used to it have essential role in the cell death. However, in recent years, we have learned that it may not be a good biomarker, as it is not that highly predict all-cause mortalities. So in our study, we used the blood phenotypic age as the primary outcomes and the telomere length as the secondary outcome. So robust research needs many lines of evidence. So to triangulate our putative findings from a drug target Mendelian randomization, we also conduct polygenic Mendelian randomization study, which means we use the genetic variants strongly predict HbA1c across the whole genome, and also conduct a cross-sectional study to assess the association of HbA1c and the aging biomarkers. We aim to verify whether the observed protective effects of metformin is explained by the glycemic trace. And we also conduct a propensity score matching to compare user of metformin with the user of other anti-diabetic drugs. So there are more than 320,000 participants of white British were included in our study with a mean age of 57 years old, and 47% were men. As we see, women have a healthy, better health profile than men. They have a younger phenotypic age and longer telomere length. So after stringent instrument selection, only 2 out of 10 encoding genes were selected to mimic the target-specific effects of metformin. And as you see, the GPD1 and AMPK gamma2 induced HbA1c lowering was associated with younger phenotypic age. And in addition, the GDP1 induced HbA1c lowering was also associated with longer telomere length. And in the polygenic Mendelian randomization analysis and the cross-sectional analysis, as we can see, the effects of HbA1c lowering was associated with younger phenotypic age, but not telomere length. And you can see their effect size is much lower than the effects of metformin-induced HbA1c. So the key message from our study, the metformin target GDP1 and AMPK gamma2 were associated with younger phenotypic age, which might partly due to HbA1c reduction. And some limitations should be considered as the ongoing investigation on the targets and mechanism of metformins. And our study might not fully capture the overall effects of metformins. And in my future study, we are planning to investigate the non-glycemic effects of metformin on healthy longevity, and we will also consider the aging clock that incorporates multi-omics, and the aging clock more organ-specific, such as brain age. So in conclusion, our study used population genetics for drug validation and repurposing, and it served as a proof of concept and provide genetic evidence that metformin have the potential benefit for healthy aging. And we add to the evidence by showing that the findings was not confounded by indication, which is a common limitation in pharmacoepidemiology study. And our study also extend the evidence that the genomic protective effects of metformin may be applied to the general population, as the UK Biobank is a population-based cohort, and metformin is the highly affordable medication with pretty nice safe profile, and we hope it is one of the sustainable strategy to promote healthy aging, and we are very much look forward to the result from the TEMP study. And lastly, I would like to thank the funder, the Healthy Longevity Costa Award from the US NAM, and the funders from the University of Hong Kong supporting in this study and supporting repurposing metformins and other drug targets for healthy longevity. And much appreciate the data source GTEx and the UK Biobank making their data available to the researcher. And also thanks my colleagues in the research team from Hong Kong U and the collaborator from Shanghai Jiao Tong University. I'll stop there. Thank you for your attention.
Thank you so much. I think maybe we have time for, there's a question down there.
Hi. Should I, as a healthy 30-year-old, be taking metformin?
I think we need more evidence, because we are, in my study, we are only capturing the effect mediated by HbA1c lowering. However, we note metformin also action on other pathway, for example, the kinase-induced GDF15, which suppress your appetite and reduce your body weight. I think we're not encouraging people taking metformin right now, but I would suggest encourage further exploration of metformin in healthy longevity.
Thank you so much.